All right. Hi, everyone. My name is Joy, and I'm a PhD student in the Azizi Lab at Columbia University. And today I'll be talking about Echidna, which is our model for mapping genotype to phenotype through joint probabilistic modeling of single-cell gene expression and chromosomal copy number variation. So the goal of Echidna is to study how copy number profile defines cancer heterogeneity and differential response to treatment. So as we all know, tumor heterogeneity is a huge problem when it comes to cancer therapy, and this is because the presence of different clones arising from genetic mutations may lead to continued resistance to treatment. So as you can see in this flowchart on the left here, over the course of treatment interventions, there's always at least one or two clones that are able to survive and go on to regrow the tumor. So there's this need for personalized therapies that are tailored for the treatment of heterogeneous clones defined by genetic mutations. So tumor clones are often characterized by changes in genomic copy number, and this refers to when the number of copies of a gene in the chromosome deviates from the expected number. So this can be through deletion events, duplication events, and so on. However, copy number variation events are difficult to detect through current bulk genome and exome sequencing, and at the same time, single-cell DNA sequencing is something that's still quite challenging to perform. So this means that any computational method developed needs to be able to make the most of the data that we do have. So the data for our project comes from melanoma patients undergoing anti-PD-1 therapy, and it consists of paired single-cell RNA and bulk whole genome sequencing data collected over multiple treatment time points. So one of our initial attempts was to apply InfoCNV, which is a method that takes in single-cell expression data and predicts the number of copies of each gene in each cell. So using these outputs, we were able to define 4 tumor clones by performing a k-means clustering and identifying cells with similar copy number profiles. So this heat map here shows the blocks of shared copy number structure among clones. And what was interesting here is that different clones, which again were defined by copy number variation alone, responded differently to treatment. So on the left here is a UMAP on cells colored by their time of collection, so either pre-treatment, on treatment, or on later. And on the right, you can see the distribution of clones. And the key observation here is that tumor clones 1 and 2 are expanding over the course of treatment. And furthermore, tumor clone 2 was enriched in immune resistance or ICR gene markers, which suggests a potentially interesting interplay between the particular copy number profile of clone 2 with immune evasion and tumor proliferation. So to backtrack a little, here we looked at copy number data and expression data side by side. But given the correlation we've seen between clones defined by copy number and expression of key genes, the relationship is clearly more complex than that. So what we'd really like is a way to integrate the two modalities and find out how one may be driving the other. And that's the goal of Echidna, to bridge genotype to phenotype in order to understand resistance mechanisms in different clones. And we achieve this through 3 main stages. So first is the integration of the two modalities through the deconvolution of whole genome sequencing. And the idea here is to leverage the resolution of single-cell RNA sequencing in order to deconvolve the whole genome data into constituent clones. Now to get those clones, we apply a Dirichlet process to assign cells based on both expression and genotype. So this is a sort of mutual optimization process that ideally results in the meaningful identification of clones, as well as the correct inference of copy number per clone. And then lastly, to bridge the gap to gene expression and phenotype, we use a hierarchical Poisson factorization process to learn cell and gene factors that may be analyzed downstream to better understand the mechanisms underlying this differential treatment response. So at its core, Echidna is just a Poisson factorization model that we derived from the SCHPF model published in 2019. So here we have our observed matrix of gene expression data X of dimensions cells by genes, which we factorize into cell factors theta and gene factors beta. And these factors share a latent dimension of D, allowing for them to be multiplied back together in order to reconstruct the expression data. So theta and beta are drawn from gamma priors, lambda and eta respectively. And there are sort of 2 things worth noting here. The first is that the eta prior is the variable we choose to interpret as the copy number of each gene per clone. And this is because eta drives the gene factors beta, similar to how we would expect copy number to influence gene expression. And then second is the addition of this clone plate here, K, which is a main deviation of our model from the vanilla HPF model. And this plate means that we have unique copy number profiles and gene factors per clone that will allow us to sort of compare dynamics between different clones. And then to incorporate the deconvolution capabilities, we introduced this variable pi, which represents the proportion of each clone in the data, and then the observed variable w corresponding to the bulk whole genome sequencing. And w can then simply be modeled as the weighted sum of the clonal proportions pi and the clone level copy numbers of each gene, which are captured through eta. And then finally, to tie everything together, we implement a Dirichlet process to assign cells to clones. And this happens through this variable zi, which represents the assignment of cell I to a particular clone. And Z influences the model in two ways. So first is through the deconvolution process we just talked about and the variable pi from which Z is sampled. And then the second is through the implementation of the clone plate. So because we have k sets of gene factors, when we take the product of theta and beta for each cell, we will only use the set of gene factors corresponding to the clone to which a particular cell has been assigned. So in this way, our model is able to integrate copy number information into the definition of tumor clones, as well as the cell and gene factors themselves, which achieves the link between genotype and phenotype. And the last thing to note here is that our model is able to integrate data across multiple time points through this outer time point plate here. And this just means that for each set of observed data, we learn unique cell and gene factors that can be used to compare dynamics across time points. However, you'll notice that eta is excluded from this plate because we don't expect the genotype of a cell to change over time. So moving on to some results, I'm starting off by showing some posterior predictive checks just to confirm that our model is fitting well to both data modalities. So here the idea is to recreate the X and W matrices in order to verify that the posteriors learned by our model are fitting correctly. And you can see that in both treatment time points, we achieve quite good recreations of both X and W. And then for the model cell assignments, which to reemphasize were defined on both expression and copy number data, you can see that our model finds two distinct clones, one of which corresponds closely to the original tumor clone that was high in immune resistance genes. So that's this clone here matching this clone over here. And this is exciting because it shows that even with expression data integrated, we still observe the same expanding clone, which supports the hypothesis that this clone is unique in both copy number profile as well as expression pattern. So diving a bit deeper, we can look at the model cell factors. So here I'm showing the factors for the pretreatment time point only. And we can see that different factors are highlighting different populations of cells. And this indicates that our model factors are able to sort of highlight varying cellular responses. And because we're more interested in the dynamics of clone 2 in particular, here in this bottom bar plot, we compute the enrichment of a particular cell factor for cells belonging to that clone. So a high value in this plot here indicates that a cell factor is enriched in clone 2 and vice versa. So moving on to the model gene factors, if you recall, we have one set of gene factors for each clone, which are shown in this top row of heat maps. And here we present the idea of defining gene modules by performing k-means clustering on the gene factors and then visualizing the resulting modules of genes by projecting the gene factors into a two-dimensional UMAP space. So to reemphasize, we are visualizing a UMAP on genes with the idea that genes with similar functions or governed by similar regulatory mechanisms will group together. So to verify this, we show the location of those ICR genes we were interested in. And as you can see, most of them are grouped together in gene module 9. And in this example, we visualize using clone 2 because, again, that's our clone of interest, but of course, you can repeat this for the other clones as well. And we can take our analyses to the next level by characterizing gene modules and cell factors together. So this heat map here shows modules defined on gene factors for clone 2, with this bar on the left indicating the distribution of modules, and this bar plot on the bottom showing the enrichment of cell factors in clone 2 as previously discussed. And so if you remember, the cell and gene factors share a latent dimension of D, which allows us to sort of look across the factor dimension to search for possible correlations. And the goal of this would be to identify cell factors that are correlated with gene modules and vice versa. So for example, if we return to module 9, which was the module where our ICR genes were localized, we can see that it is high in this factor dimension of 6. And this also corresponds to a cell factor that was enriched in clone 2. So then what we can do is perform pathway analyses such as David and GSEA. So for a given gene module, we can run a David gene ontology analysis by inputting all of the constituent genes of a module in order to highlight potential pathways associated. So you can see some results on the left here. And then for cell factors, we can perform gene set enrichment analyses by ranking genes based on their correlation to the cell factor. And I won't go into too much depth on the biological analyses here, but just to highlight a few key results. For the cell factor that is associated with the expanding clone, factor 6 here, we do see enrichment in cancer-associated pathways such as EMT, TNF-alpha, apoptosis, and so on, which confirms the interpretability of our model. And the purpose of these analyses is to just help answer the question of what functions the highlighted modules are associated with, and how that may be tied to the dynamics of the cell factors of interest. And so the other sort of analyses our model can perform is the comparison of factors across different clones in order to characterize tumor heterogeneity. So just to illustrate, here we have gene factors in clone 1 compared to in clone 2, both in the pretreatment time point. And here I've marked the modules where the ICR genes are localized. So that's module 6 in clone 1 and module 9 in clone 2. And you can see that in clone 2, the ICR module is corresponding to cell factors associated with the expanding clone. Whereas in clone 1, if we look across all of the factors here, we can see that the module is only enriched in factors that are not associated with the expanding clone, which raises the hypothesis of whether or not immune resistance is only tied to tumor cells undergoing active proliferation. And then just like we can compare factors between clones in the same time point, we can also compare across time points to characterize differential response to treatment. So here we're showing the factors in clone 2 in the pre-treatment time point and in the on later treatment time point. And we can do something as simple as comparing the set of enriched pathways for key cell factors as shown below, or we can repeat all of the analyses previously shown to get an idea of how gene pathways and cellular dynamics differ with treatment intervention. So these are all examples of the types of analyses that can be done with our model outputs. So our work is still ongoing, and one of the last pieces of the puzzle that we've been working on is optimizing our deconvolution of the whole genome sequencing data. So here are some preliminary attempts at deconvolving individual time points. So in each heat map, we are comparing our model ADAs, which is the top row, to the InfraCNV results as a sort of ground truth. And you can see that for at least some of the clones, we get quite good recreation of the InfraCNV results. And then on the right, we have the same posterior predictive check on W, just to confirm that we are still able to fit to the whole genome sequencing data as well. So I thought it would be fitting to end on this note of deconvolution, because this is really what sets our model apart. Every aspect of our model is impacted on some level by the inferred copy number states, whether it's through driving the choice of gene factors or the assignment of cells to clusters. So future analyses will involve looking at these inferred copy number profiles in conjunction with the defined gene modules to truly achieve a sort of holistic view of how clonal genotype impacts phenotype and the efficacy of cancer therapies. So here are our acknowledgements and references, as well as our funding sources we'd like to thank. Thank you all for your attention, and I'll take any questions.
Very nice work. I was curious, how easy is it to do inference in this model, and in terms of computational complexity and things like that?
Yeah, so the runtime of our model is it's not bad. So we run using a GPU, and it takes probably like several hours to perform. So compared to like InfraCNV, which takes multiple days to do the inference on the single cell level, our model is much faster.
Can you say, how do you set the hyperparameters of the model? You mentioned like a genotype process. So is there a hyperparameter with that, and how is that set?
Yeah, so the hyperparameter is just through a lot of fine tuning and sort of trial and error. So for the Dirichlet process, for example, we set the parameters to ensure relatively even distribution of cluster sizes, for example, and that's just through a lot of manual fine tuning.
Okay, thank you.
Thank you.